Hello. We are here today at the last day of CROI in Boston 2014, the Conference on Retrovirus and Opportunistic Infections. And we have two speakers today who've been very generous of their time to be with us this morning in the busy conference life. Nicolas Chaumont is assistant member and principal investigator of the Vaccine and Gene Therapy Institute in Florida. He's working to define mechanisms of potential drug targets that could lead to the eradication of persistent HIV and an eventual cure for HIV infection. And he's an investigator with the DARE Collaboratory from the Martin Delaney Collaboratory. And Sharon Lewin is here with us again this year. She's a professor and head of the Department of Infectious Diseases at Monash University and Alfred Hospital, co-head Center for Biomedical Research at the Burnett Institute. And she's also the AIDS 2014 co-chair of the International AIDS Conference coming up very soon. And she's also an investigator with the DARE Collaboratory and the Martin Delaney Collaboratory. So thank you so much for being here today. I just wanted to start out by saying it's a very busy year, a busy conference. There's some good news, some sort of maybe sobering news, but I think CROI was very careful this year in selecting presentations that may not have been as maybe earth-shattering as the Mississippi Baby, so they were a little more careful about this year. But I kind of think that we're in a phase and a time where the progress in cure research is really like the early days of development of HIV drugs. We're still at such an early phase where we're trying to figure out even the very early basic questions. And so, Nicolas, since you do a lot of work with viral persistence in that area, I wondered if you could give us an update.
Sure, absolutely. So I think what we're starting to realize is that these questions of viral persistence are probably much more complex than we originally thought. So 10 years ago, we were thinking that all CD4 cells in which the virus mainly persists were pretty much the same, and that the virus was persisting in all these cells in the same way. And I think what we've learned during the past few years, and that we still learn today, is that this complexity is actually much more profound than we thought before. And two reservoir cells are never the same, so they're quite different. We've learned that in the past from past studies from Jason Brenchley group. Then my lab also worked on this question. And at this conference, we had a nice presentation from Matthias Lichtenfeld, who was actually able to demonstrate that there are other cell subsets, and this particular cell subset that we call the stem cell memory cells, in which the virus can also persist for a very long period of time. And the implication for this finding is that it's probably not with a single strategy that we're going to be able to get rid of these cells, because they all behave in a very different way, in a different manner, and the virus persists probably in different ways also in these cells. So I would say that's probably, you would say that it's bad news because it adds more complexity to the issue of persistence, but I would say that it's also good news because now we are really trying to get the big picture and what we should do to get rid of these cells and to get rid of this persistent HIV. So that's on the description of the reservoir. The other thing is that we're getting better tools and better assays to measure HIV persistence. Sharon and I have been struggling for a very long time with the different assays we can use to measure HIV persistence in people who have been receiving ART for many years. The frequency of these cells, as you know, is extremely low, and we need to use very sensitive assays. At the same time, these assays have to be very specific. And the assays that we had in the past were working quite well, but we needed like tons of cells, like millions of cells to be able to run them. They were expensive, cumbersome, took a lot of time. There are more and more alternative assays that are developed these days, which I think will probably allow us to move much faster than we did during the past 10 or 15 years. So this is really a good news, definitely a good news. And yes, and that's it.
Okay. Where are you in your work in Miami?
So I work in collaboration with Sharon. So as you said, we are part of this DARE collaboratory. Yes. So the idea of DARE is to really look at the interaction between HIV persistence, so the reservoir, and the immune system, and how the immune system can either contribute or, on the contrary, maybe reduce this level of HIV persistence. So what I'm working on is I'm trying to identify biomarkers of those cells that persist.
Right.
And that's the work that I'm doing with Rémi Fromentin. And we're trying to identify markers expressed at the surface of these cells and manipulate those markers to eliminate those latently infected CD40 cells. So that's a collaboration that we have, again, in the context of there, with Steve Dix, Rafik Sekali, Rémi Fromentin, Sharon, many people involved in that. I think the important aspect of this collaboration is that we're sharing samples. So we have this ability to really get the big picture by measuring many, many parameters in a limited number of subjects and trying really to take what's the most important parameter for these cells to survive.
And this is sort of highlights the really important need to collaborate, and the collaboration process that the Martin Delaney cohort has set up is really critical. Okay. So in terms of the biomarkers and the assays, how far along? When do you think we'll have the best assay? When is everybody going to agree? Where are we with that?
It's going to be tough to reconcile everybody on these questions, because basically every single lab working on these questions of HIV persistence has its favorite assay, and it's always easier to say I have the best. Absolutely, right now, actually, it's easy to say that because we never really formally compare all these assays together. Even though there was a study published last year by a group of Janet Tilicano, this was an unlimited number of subjects, and all the different assays were not included in that study. So I think we need to do a larger study that will really look at not only the capacity of this assay to measure something that can be reproduced by others, but also to their robustness, I would say, if those assays can be repeated on the same samples and would give the same result. So there is a lot of progress that is being made right now on these questions. I think there's probably going to be a lot of collaborations with the goal to identifying the best assay that we can have to measure HIV persistence.
And does the FDA have to come in and then standardize the assay? Is that
well, eventually that's probably something that will happen. At the same time, I think it's important to still develop novel assays and not say, okay, we have this assay, we'll keep it like that, and that's the universal assay that we're going to use for any question on HIV persistence. I think that would be wrong to do that. I think depending on the questions we're asking, we might want to use one assay or another. I think we still have a lot of work in the lab to do to validate this assay before we get any validation by the FDA.
Can you give any kind of timeline?
I can't.
Yeah. We're always wanting to know the timeline. Yes.
That's a very difficult question. What I can tell you is that during this past year, there are many assays that have been developed, and I think this is really something new because we've been working with the same assay for like 10 or 15 years, and now within the last year, I have been at the top of my mind almost like 4 or 5 different new assays that are completely different from the one that we had in the past that have been developed.
So more and more science is developing to create those different assays, and everybody's competing, and Okay. Well, so Nicholas has to chair a session this morning at 10 o'clock, so we're going to break away, and we'll be right back. Thank you. Welcome back to the IFARA Cure report back from CROI 2014, and I wanted to bring in the topic of the Cure Baby, Mississippi Cure Baby, that was presented last year at CROI, and we had another report from Deborah Persad this year, and I wondered if Sharon, you could give us a little update on that.
Yeah, so Deborah first of all gave us an update on the Mississippi Baby. Now 24 months off treatment, no rebound virus, but actually found traces of virus in the baby cells. So I think there was a little controversy early on about was the Mississippi Baby indeed infected, but what she showed us I think confirms that baby was, and this is truly a remission for the Mississippi Baby or functional cure.
And can you explain that a little bit about finding that residual virus, because they also found it in Timothy Brown.
Yeah.
So
that's right. In Timothy Brown, it was much lower levels, and what you can do when you find the virus using a very fancy test that can detect very low levels of virus. And anyone that works in the lab using this sort of test, what's called a PCR test, you're always worried, is this laboratory contamination, because the test is so sensitive. And I think with Timothy Brown, the jury's not out. Was this real or was this contamination? I think with the Mississippi baby, it's a little different because they found much higher levels of persistent DNA or traces of virus in multiple cell types. And they've also done some sequencing and show that it's not a laboratory contaminant.
Okay.
So I think it's a little different. She then told us about this new baby from California, near Los Angeles.
Los Angeles.
And this baby was delivered actually shortly after CROI. So I think that was informed the doctors to perhaps to then start immediate treatment on this baby at 4 hours following delivery. Baby was confirmed as infected using the standard tools that paediatricians use, which means 2 positive tests. In fact, the baby also had evidence of virus in their spinal fluid before they started treatment at 4 hours. Started full dose antiviral treatment, again what the Mississippi baby had, and detected virus shortly after, but then no detectable virus in blood, but detectable virus in cells or DNA. Baby's been on treatment for 8 months and hasn't yet stopped treatment. So I think it's a little early to even say this baby is in remission or it's a functional cure.
I mean, it's one of the questions that we have in some of these studies is that we have to really stop treatment to see if it's valid.
Yeah, you need to stop treatment. And of course, we learned from the Boston patients, we stopped treatment, a lot of excitement at 8 and 15 weeks, but then the virus came back.
Can you expand upon that a little bit? Because that was a big report at this conference. It was reported earlier, but it was reported again here.
Yeah, so the Boston patients were 2 men who had HIV, were on antiviral therapy and had a lymphoma and needed a bone marrow transplant for their lymphoma. And it's just like the Berlin patient, they received a transplant. But the difference here was, first, that the bone marrow that they received was from a regular donor. Nothing special about being resistant to HIV, which is different to the Berlin patient.
Right, and people get that confused, I think.
Yeah, the Berlin patient received a unique type of donor bone marrow that came from someone that was naturally resistant to HIV. The Boston patients received a bone marrow that was not resistant. The Boston patients also didn't have, mega irradiation therapy. They had sort of modified irradiation therapy. And but the Boston patients stayed on their anti-HIV drugs, antivirals, during the transplant and for 2 and 4 years after the transplant. And what the investigators, Tim Henrik and his team, found was that when they measured HIV in cells, which is the HIV DNA test, within 6 months that was cleared from the Boston patients. They tried to grow virus. They couldn't grow virus. They looked in one patient in the gastrointestinal tract, couldn't find virus. So after, I think it was 2 and 4 years on treatment, they did the ultimate test, which is stopping antiviral drugs and seeing if the virus came back. And there was a short window where things looked very exciting because it appeared that these patients were in remission. But as we've learnt before with HIV, there's always these unexpected findings, and the virus is tricky, and the 2 patients eventually did rebound at 8 and 15 weeks after stopping treatment. Now that is much longer than we normally see viral rebound. We normally see, when people stop treatment, the virus normally takes about 3 weeks to come back. So it took longer for the Boston patients, and we think that's a indirect measure of how much virus was left. If you've got a tiny amount of virus, it may take longer to come back.
Okay, so that in itself is an advance.
Yeah. I think that was a big advance, knowing that. And also, but the Boston patients are also a little different to regular patients who might stop their drugs, and that is they had a new immune system from their transplant, and the new immune system had never seen HIV before.
Okay.
So when the virus did come back, it was like a primary infection, and the virus went from undetectable to very high levels very quickly.
Yeah,
and that's
interesting.
probably because the Boston patients were effectively like patients that had never been infected before.
And do you know where Heinrich is going with this research? Do you know what his next steps are?
Yeah, I think he's really going to dissect the immunology very, very carefully. You know, what were, we always think about T cells as being the major cell involved in keeping the virus under control. But there are many other immune cells that could be important, one particularly called natural killer cells. And maybe it's their immune footprint, or what we call HLA type, that was really relevant. So I think he's going to go into a lot of detail around the immunology and also the virology. If you look at you can sequence the virus that popped back up. Where did it come from? To try and find out where did that virus come from? Why couldn't we find it in the blood using our routine tests?
Are they going to look at other patients?
Yeah, I think everyone around the world is trying to find their patients. We have a patient, in fact, who received a allogeneic transplant under continuous antiretroviral therapy late last year, and we're still characterising that patient. But I think cases of the Mississippi baby, cases of the Boston patients stimulate a lot of new ideas and new research. And these are rare cases, but it means everyone goes home and starts looking for them in their clinic. And if we can characterise more of these cases, I think we're going to learn a lot more.
So I think one of the questions that comes up around these transplant issues is that these are patients that are very sick with some kind of malignancy. So what is the next step beyond studying those particular types of patients? Or do we know?
Yeah, I mean, I think there are different ways to, there are different ways that you can clear out T cells than just by transplantation. You can use some immunomodulatory treatments.
Sure.
And that could be of interest. I think developing animal models where you can mimic this, and there is some work going on in non-human primates.
Yes.
Which allows you to manipulate the system a lot more easily, and they may give us clues. Because you're right, we're looking in a population of people who have underlying lymphoma and underlying immune defects.
Yeah, and so I guess this kind of highlights that we're just still really very early in understanding this whole platform, this whole sort of pathway towards looking at remission.
Yeah, I think many investigators I think clinical trials and careful observational studies are really important because they generate new ideas and new lines of thinking. But I think all of us just keep coming back to the fact that we need to understand the basic biology a lot more to really tackle the problem and get new targets and approaches.
Yeah, the early answers lead to more questions.
Yeah,
so.
yeah.
Would you like to talk a little bit about the upcoming World AIDS Conference? Since
Sure,
you're the co-chair?
So we're not far away, July, so 5 months to go.
Exciting.
We're expecting, we hope, 14,000 people to come to Melbourne. It will be the first time the conference has ever been in Australia, and the first time in 10 years that it's back in the Asia-Pacific region. So there's going to be a lot of attention drawn to the kind of epidemic that we see in our region, which is still an epidemic in key affected populations, and an epidemic where we aren't seeing the great success stories that many other parts of the world are seeing. We're seeing increasing infections in men who have sex with men, people who inject drugs, sex workers. Unfortunately, a lot of very discriminatory laws that we still see in our region and elsewhere against gay men and groups like injecting drug users, and how that's extremely unhelpful for getting public health messages. Not just that, it's unacceptable, but for a whole range of reasons, but it certainly worsens the fight against eliminating new infections. There's going to be a lot of interesting cure work. I'm co-chairing the conference with Françoise Barré-Sinoussi, the president of the International AIDS
Very busy
Society.
woman these days.
Yeah, and Françoise has really spearheaded this whole galvanised the scientific community around cure. So it's a great pleasure to co-chair with Françoise. We're going to have a cure workshop on the Saturday and Sunday before the meeting.
Yes.
And we're actually going to tap into some great expertise that we have in Australia in the oncology and immunology field. Australia's had some really great research for a long time, particularly in immunology. So we've invited investigators that don't work in the HIV field to talk about graft versus world expert Jeff Hill to talk about graft versus host disease. Another expert, Miles Prince, who did a lot of the early phase 1, phase 2 studies of HDAC inhibitors. We work with Miles on our varinostat study, but he's going to give a plenary on the field of epigenetic regulation, or how you manipulate genes in cancer, which is very relevant to HIV. So I think, you know, great scientific advances often come left field or from outside the discipline, and we need to look more.
And again, this is where the collaboration comes in, and bringing in people from different fields and having that discussion, I think, is critical. So, well, we're excited to be having.
It's going to be a lot warmer than Boston.
Good.
It's winter, but it will be a lot warmer.
Yeah.
And actually, the city is getting really excited. It's amazing. Melbourne's
Great.
a very nice city. The conference centre is right in the centre of town. And there's going to be a lot of great art and music and sport and culture and a whole lot of great stuff. Painting the town red for the week.
Good.
That's the plan.
Okay. Well, thanks, Sharon, very much for being here today, and
Pleasure,
appreciate
as
it.
always.
We'll talk to you next year.
Okay, great.
Okay.
Thanks.